Welcome to the Deep Dive, your shortcut to understanding complex topics. Today, we're diving into the incredible world of the urinary system, a, uh, a real marvel of engineering tucked away inside us.
It really is.
We're drawing our insights from Medical Physiology by Boron and Boulpaep, and our mission really is to break down this dense material into clear, engaging, and clinically relevant insights so you can really grasp why this system is so vital.
It's truly remarkable. Did you know your kidneys, I mean, they're less than 0.5% of your total body weight, right?
Yeah, tiny.
But they actually filter about 1/5 of your heart's entire blood output every single minute.
Wow, 1/5 every minute.
Every minute. That's just an astonishing amount of fluid they process.
That really puts into perspective just how hardworking these organs are. So, okay, what exactly is their job? Beyond just filtering, what are maybe the big three functions we should keep in mind as we explore?
That's a great question. It's easy to think of kidneys as simple filters, but honestly, that's just one piece of the puzzle. First, yes, they are incredible filters, constantly removing metabolic waste products, toxins, stuff like that from your blood.
Right, the cleanup crew.
Exactly. Second, they're precise regulators. They're tirelessly maintaining your body's fluid levels, electrolyte balance, sodium, potassium, and acid-base balance. Super important.
Okay, regulating the internal environment.
Precisely. And third, something often overlooked, they act as powerful endocrine glands. They produce or activate crucial hormones that influence, well, everything from red blood cell production to calcium metabolism and even your overall blood pressure.
Endocrine glands too. Okay, that's more complex than I thought. Let's unpack this macroscopic masterpiece then. When you picture your kidneys, what are we looking at?
Okay, so think of two bean-shaped organs. Each one's about the size of, say, a large bar of soap,
Okay.
weighing around 125 to 170 grams for men, a bit less for women. And they're nestled behind the peritoneum, basically resting on each side of your spine, kind of from your lower ridge down towards your upper pelvis.
Retroperitoneal, right, behind the abdominal lining.
Exactly. And each kidney is wrapped in this tough, protective, fibrous capsule. It doesn't stretch much, which helps keep everything sort of contained.
Secure. And what about where things go in and out?
Right. If you look at the inner curve of that bean shape, you'll find a central indentation called the hilus.
The hilus.
Yeah, think of it as the kidney's main entrance and exit gate. This is where the renal artery brings blood in, nerves enter, and then the renal vein, lymphatic vessels, and the ureter, that's the tube carrying urine away, all depart.
Got it. A busy doorway.
Very busy. And the hilus opens into a small space called the renal sinus. This just holds the renal pelvis, which is like a funnel.
Funneling the urine.
Exactly, collecting urine and funneling it into these little cup-like extensions called calyces, major and minor calyces.
Okay. Now, if we were to slice a kidney open, what would we see inside? Layers.
You have two distinct layers. There's the outer granular region, the cortex. It looks kind of speckled.
Speckled. Why?
Because it's densely packed with millions of tiny microscopic filtering units. The glomeruli and all these winding tubules, that gives it that granular look.
Okay, cortex on the outside. What's inside?
Then you have a darker inner region, the medulla.
Medulla.
And contrast to the cortex, the medulla looks more striped. It doesn't have those initial filtering units, the glomeruli. Instead, it's characterized by these parallel arrangements of tubules and small blood vessels.
Stripes.
And within the medulla, you'll see about maybe 8 to 18 cone-shaped structures. These are the renal pyramids.
Pyramids.
Yeah, and the tips of these pyramids, called papillae, they act like tiny little spouts, dripping the newly formed urine directly into those calyces we mentioned.
Dripping into the funnel system. Okay, I'm picturing it. So this incredible structure needs a pretty unique plumbing system, I imagine.
Absolutely. And here's where it gets really interesting, tying back to that blood flow fact.
Right, the 20%.
Yeah. Despite making up less than 0.5% of your body weight, your kidneys demand an astounding 20% of your entire cardiac output.
It's just wild.
It is. And that massive amount of blood flow is absolutely crucial for their main job, filtering your blood plasma.
So how does the plumbing work to handle that?
Well, they achieve this through a highly specialized vascular sequence. Blood enters each kidney via a single renal artery, right?
Okay.
Which then branches and branches and branches again, eventually leading to this unique two-arterial, two-capillary bed system.
Two arterioles. That's unusual.
Very unusual. Think of it like this. An afferent arteriole, which is a high-resistance vessel, brings blood into the glomerular capillary network. This network is under high pressure, perfect for filtration.
Okay, push the fluid out.
Exactly. Then, instead of going straight to a vein, the blood flows out through another high-resistance vessel, the efferent arteriole.
Afferent in, efferent out, both arterioles.
Correct. And this efferent arteriole then leads into a second low-pressure capillary network called the peritubular capillaries. These wrap all around the kidney's tubules.
Ah, so the first capillary bed filters, the second one reabsorbs.
Precisely. It facilitates reabsorption of all the useful stuff the kidney wants to keep.
And the brilliance of having those two arterioles, what's the point?
The control. Because they're arterioles, they can constrict or dilate. And this ability, which is tightly controlled by nerves and chemical signals, directly determines the pressure inside those filtering glomerular capillaries.
So the kidney can fine-tune its own filtration pressure.
Exactly. It allows the kidney to precisely regulate how much blood it filters, moment by moment.
Yeah.
It's really quite elegant.
Okay. And are all these capillary systems the same throughout the kidney?
Not quite. There's
No.
a bit of specialization. For most of the filtering units, those near the surface,
The superficial ones?
yeah, the superficial glomeruli, their efferent arterioles form that dense network of peritubular capillaries supplying the surrounding tubules in the outer kidney, the cortex.
Okay.
But for a special group of filtering units located deeper, right at the border between the cortex and medulla.
The juxtamedullary ones.
You got it. The juxtamedullary glomeruli. Their efferent arterioles do something different. They dive deep down into the medulla, forming these long, hairpin-shaped vessels called the vasa recta.
Vasa recta, hairpin loops.
Yep. And these are critical for supplying the tubules way down in the deep medulla. It's also worth remembering, blood flow isn't equal. About 90% goes to the superficial part, the cortex.
And only 10% to the deep medulla.
Roughly.
Yes.
Which is important for how the medulla functions.
And speaking of precise design, you mentioned lymphatics earlier. Draining fluid, are they everywhere?
Good point. Lymphatics are present in the outer kidney, the cortex, draining interstitial fluid like they do elsewhere. But, and this is key, they're noticeably absent from the deeper inner medulla.
Why? Why leave them out there?
That's a fantastic question, and it's absolutely by design. Their absence in the medulla helps preserve the extremely high concentration of solutes, salts, and urea, mostly in the interstitial fluid there.
The osmotic gradient.
Exactly. This hyperosmotic environment is absolutely necessary for the kidney's ability to produce concentrated urine later on. If lymphatics were there, they'd just drain away that critical gradient and poof, no water conservation.
Clever, deliberate omission. Okay, so all this amazing anatomy, the layers, the blood flow, it all serves the kidney's true functional powerhouse, the nephron.
The nephron, that's the unit. Each of your kidneys is home to an astonishing number, maybe 800,000 to over 1 million of these tiny independent processing units.
1 million per kidney. Wow.
Yeah, each nephron works largely by itself until its very end, the collecting duct finally merges with others to pool the urine.
So what makes up one nephron? What are the parts?
Okay, the basic structure has two main parts, the glomerulus and the tubule.
Glomerulus, that's the filter bit, right?
That's the filter bit, the dense tangle of blood vessels where the plasma first gets filtered. Then you have the tubule, which is a long, winding epithelial tube. Its job is to take that initial filtrate and process it, turning it into final urine.
So filtering, then processing.
Exactly. And these two systems, the vascular part, glomerulus, and the tubular part, meet at a really critical point, Bowman capsule.
Bowman's capsule.
Yeah, it's like this cup-shaped structure, basically the blind end of the tubule, that completely surrounds the glomerulus. It creates a space inside, Bowman space, where the filtered fluid actually collects. This is where filtration truly begins, where fluid leaves the blood and enters the tubule system.
Okay, the interface. And you mentioned different types of nephrons earlier, superficial and...
Right. Two main populations.
Yeah.
You have the superficial nephrons, which have relatively short loops of Henle, that's a section of the tubule, that only dip a little way into the medulla.
Short loops.
Then you have the juxtamedullary nephrons, and these are incredibly important.
Why are they so important?
Because these juxtamedullary nephrons have much longer loops of Henle that dive deep, sometimes all the way down to the very tip of the medulla.
Deep loops.
Deep loops. And this deep penetration is absolutely crucial for the kidney's ability to produce concentrated urine, which, as we said, is vital for conserving water.
Makes sense. Longer loop, more interaction with that concentrated medulla.
Precisely. It's also worth just a quick mention that these complex structures, they develop through this fascinating back and forth interaction between two different embryonic tissues, leading to their final intricate shape.
Wow, development's always amazing. Okay, so we have the nephron parts, let's talk about that filtration barrier itself, the gatekeeper.
Right, the ultimate
Yes.
gatekeeper. This happens in the renal corpuscle, which is just the term for the glomerulus plus Bowman's capsule. This is the spot where that initial glomerular filtrate is formed.
And it's not just one layer, is it?
No way. The brilliance here is that this barrier is made up of 4 distinct layers, each working together like a multi-layered security system. Really smart filter.
4 layers. Okay, walk us through them. Layer 1.
First, covering the inner surface of the blood vessel cells themselves, you have the glycocalyx. Think of it like a sticky, negatively charged coating, like a velvet rope, maybe.
Okay, a charged barrier.
Exactly. It prevents large, negatively charged molecules, like some proteins, from even getting close to the actual filter pores.
Smart. Layer 2.
Then you have the endothelial cells themselves, the cells forming the capillary wall. These cells are perforated by these relatively large pores, or fenestrations, about 70 nanometers wide.
Fenestrations. Like little windows?
Kind of.
Mm.
Like a colander, maybe. They let water and small solutes, salts, glucose, amino acids pass through easily. But crucially, they block larger elements like your red blood cells and white blood cells. Keeps them in the blood where they belong.
Okay, cells stay in. Layer 3.
The third layer is the glomerular basement membrane, or GBM. This is a specialized mesh, sort of like felt, sandwiched between those endothelial cells in the next layer we'll talk about.
Basement membrane. What does it do?
It has multiple sublayers itself and acts like a much finer sieve. It restricts anything over a certain size, particularly intermediate to large solutes. But importantly, it's also packed with negatively charged components called heparin sulfate proteoglycans.
More negative charges.
More negative charges. So it strongly repels any large, negatively charged proteins that might have gotten past the glycocalyx. It's a double whammy against letting protein escape.
Very selective. Okay, what's the final layer? Layer 4.
Okay, this one's fascinating. The podocytes.
Mm.
These are highly specialized cells, technically part of Bowman's capsule epithelium, but they look totally different. They have these incredible branching foot processes, like little fingers.
Foot processes.
Yeah, pedicels is the technical term. And these foot processes meticulously wrap around the outside of the basement membrane, covering the capillaries. But they don't form a solid sheet, they interdigitate, like clasping fingers.
Okay, so there are gaps between them.
Exactly. Between these interdigicating foot processes are tiny gaps called filtration slits. And these slits aren't just empty space. They're bridged by a delicate structure known as the slit diaphragm. Think of it like a final, ultrafine mesh with tiny pores only 4 to 14 nanometers wide.
Wow, super fine.
Super fine. And guess what? The podocytes, the slits, the slit diaphragm, they're also covered in negatively charged glycoproteins.
Adding even more negative charge barrier.
Yes, it's a multi-pronged defense, particularly against losing valuable proteins like albumin from the blood. Size exclusion and charge repulsion working together.
So if that delicate structure, especially the slit diaphragm, gets damaged.
Then you can have big problems. For instance, there's a condition called Finnish-type nephrosis. It's a genetic disorder where a key protein in that slit diaphragm called nephrin is missing.
And what happens?
Massive proteinuria. Huge amounts of protein leak out of the blood and into the urine because that final barrier is defective. It really highlights just how critical this intricate, multi-layered structure is. It's a guardian, not just a simple sieve.
Absolutely. A very sophisticated guardian.
Mhm.
And you mentioned other cells nearby, mesangial cells.
Right. Supporting these delicate glomerular capillary loops nestled between them is a network of contractile cells called mesangial cells. They provide structural support, secrete the matrix around them, and can actually contract to influence filtration surface area. They're kind of like the groundskeepers of the glomerulus.
Okay. All these components work together. Which brings us, I think, to one of the kidney's key control centers you mentioned earlier, the juxtaglomerular apparatus, or JGA.
The JGA, yes.
Yeah.
Think of it as the kidney's own internal thermostat, or maybe a quality control checkpoint, constantly monitoring and adjusting.
Where exactly is it?
It's a specialized region formed right where a part of the nephron's tubule, specifically the final part of the loop of Henle, the thick ascending limb, loops back and comes into direct physical contact with its own glomerulus, particularly the afferent and efferent arterioles.
The tubule talks to its own filter.
Exactly. It's all about feedback. The JGA is composed of a few key cell types working together. First, the macula densa. These are specialized epithelial cells in the wall of that tubule right where it touches the arterioles. They act like chemical sensors.
Sensing what?
They sense the concentration of sodium chloride, essentially, in the fluid flowing past them inside the tubule.
Okay, salt sensors. What else?
Then you have the granular cells, also called juxtaglomerular or JG cells. These are specialized, smooth muscle cells located mainly in the wall of the afferent arteriole, right near the glomerulus,
And they're granular
because they contain granules filled with an enzyme called renin. These are the cells that actually produce, store, and release renin into the bloodstream.
renin. Okay, that sounds important. Are there other cells?
There are also the extraglomerular mesangial cells, which are basically continuous with those mesangial cells inside the glomerulus, sitting in the angle between the arterioles and the macula densa. They seem to help relay signals.
So what does this whole JGA setup do in terms of regulation?
Well, it's central to two major regulatory functions. First, it's responsible for tubular glomerular feedback, or TGF.
To be talking to the glomerulus.
Exactly. If the macula densa senses that too much salt is flowing past it in the tubule fluid, which usually implies the filtration rate was too high.
It tells the glomerulus to slow down.
Basically, yes. It triggers signals, likely involving ATP release, that cause the afferent arteriole feeding that same glomerulus to constrict. This reduces blood flow into the glomerulus, lowers the filtration pressure, and brings the filtration rate back down. It's brilliant intrinsic autoregulation for each individual nephron.
Wow. Local control. What's the second function?
The second major function is controlling renin release.
From those granular cells.
Right. The granular cells act like pressure sensors, or baroreceptors. If they sense a decrease in stretch in the afferent arterial wall, which could happen if your overall blood pressure drops, for instance,
They release renin.
they increase the release of renin. Also, sympathetic nerve stimulation strongly triggers renin release, and signals from the macula densa, specifically low salt, can also increase renin release.
And why is renin so important?
Renin is the very first and rate-limiting step in the crucial renin-angiotensin-aldosterone system, or RAS.
Ah, the RAAS. Heard of that.
Yeah, it's a major hormonal cascade that ultimately leads to increased sodium and water retention by the kidneys and vasoconstriction throughout the body, which works to raise your systemic blood pressure. So the JGA plays a key role in both short-term, local control of filtration and long-term control of your overall blood pressure.
It's all incredibly connected. Now, you mentioned sympathetic nerve stimulating renin. Are the kidneys heavily innervated otherwise?
Oh yes, the kidneys receive extensive sympathetic innervation, but interestingly, no significant parasympathetic input.
Just sympathetic. What does that do?
Sympathetic nerves primarily cause vasoconstriction of the renal blood vessels, especially the afferent arterioles. They can also directly enhance sodium reabsorption by the proximal parts of the tubule, and as we said, they strongly stimulate renin secretion from the JGA. Generally, sympathetic activity decreases renal blood flow and filtration, conserving fluid during stress, like fight or flight.
Okay, are there sensory nerves too? Do the kidneys send signals back?
Yes, they do. They have afferent or sensory nerves. Some act like internal pressure gauges, baroreceptors, located in the larger arteries and afferent arterioles, responding to changes in perfusion pressure.
And others?
Others are chemoreceptors, found mainly in the renal pelvis, near where urine collects. They seem to respond to things like ischemia, lack of oxygen, or abnormal ion composition in the urine, like high potassium or acid. These signals might feed back to influence local blood flow or perhaps signal pain, like in kidney stones.
Fascinating. Okay, beyond renin and regulating blood flow, what about those other endocrine roles you mentioned way back at the start?
Right. Kidneys do more hormonally. A big one is vitamin D activation.
Vitamin D, I thought that came from the sun.
Well, sunlight helps your skin make a precursor, but that form isn't active. Cells in the proximal tubule of the nephron perform the final critical step, converting circulating 25-hydroxyvitamin D into its fully active form, 1025-dihydroxyvitamin D, also called calcitriol.
And why is active vitamin D important?
It's absolutely critical for regulating calcium and phosphorus absorption from your gut and for maintaining healthy bone structure. Without the kidneys doing this final activation step, you'd have serious problems with calcium balance and bone health.
Okay, so kidneys are key for bones too. What else?
They'd also produce erythropoietin, or EPO.
EPO, the red blood cell hormone.
That's the one. Specialized fibroblast-like cells, mainly located in the cortex and outer medulla interstitium, sense the oxygen levels in the surrounding tissue. If oxygen levels drop, hypoxia,
They release EPO.
they secrete EPO into the blood. EPO then travels to your bone marrow and stimulates the production and development of red blood cells, increasing your blood's oxygen-carrying capacity.
And this is clinically huge, right, for kidney disease?
Absolutely massive. In chronic kidney disease, as the kidneys fail, EPO production drops significantly. This leads to severe anemia, causing fatigue, weakness, all sorts of problems. Being able to give patients recombinant EPO, synthetic EPO, has been a complete game changer, dramatically improving their quality of life.
Incredible impact. Are there any other, maybe more local hormones?
Yeah, the kidneys also release various local signaling molecules, paracrine factors, things like prostaglandins and kinins, which often act as vasodilators, potentially playing a protective role by counteracting excessive vasoconstriction during periods of reduced blood flow. There are others too, like angiotensin being produced locally, bradykinin, even ATP acting as signals, but their exact local roles are still being fully worked out.
So much going on. Okay, how on earth do we actually measure all this incredible function? If you go to the doctor, how do they check your kidney function?
That's where the power of clearance comes in. It's a fundamental concept in renal physiology.
Clearance. What does that actually mean, clearing something out?
Sort of, but it's a bit more abstract. The clearance of a substance is defined as the virtual volume of plasma that would be totally cleared or emptied of that specific substance in a given amount of time, usually per minute.
Virtual volume. Okay, explain that.
Right. Imagine your kidneys excrete, say, 0.14 millimoles of sodium per minute. And let's say the concentration of sodium in your plasma is 0.14 millimoles per milliliter. To get that amount of sodium out, the kidneys would have needed to completely clear 1 milliliter of plasma of all its sodium.
Ah, even though much more plasma actually flowed through.
Exactly. Maybe 700 milliliters of plasma flowed through the kidneys that minute, but only the sodium equivalent to 1 milliliter was removed. So the clearance of sodium in that case would be 1 ml a minute. It's the volume effectively cleared.
Okay, I think I get it. It's a rate of removal expressed as a volume. How do we calculate it?
The classic clearance equation is pretty straightforward. Based on the principle of mass balance, what goes in must come out or be accumulated. Clearance of substance X, written as Cx, equals the concentration of X in the urine, Ux, multiplied by the urine flow rate, V dot, all divided by the concentration of X in the plasma, Px. So Cx at Ux, Vg, Vx.
Okay, urine concentration times flow rate divided by plasma concentration.
Yep. And this value can tell us a lot. For substances like glucose, which is normally completely reabsorbed back into the blood by the tubules.
Its urine concentration is 0, so clearance is 0.
Exactly. Clearance is 0. But for something like, say, p-aminohyppuate, or PAH, a substance that's not only filtered but also actively secreted into the tubule so efficiently that it's almost entirely removed from the blood in a single pass through the kidney.
Its clearance would be really high.
Very high. Its clearance approximates the total renal plasma flow, RPF, which is typically around 600, 700 milliliters per minute.
Wow, big range. So we can use specific substances to measure specific things.
Yes, precisely. We use PAH clearance to get a good estimate of the kidney's overall renal plasma flow, RPF.
Okay. And what about the filtration rate itself? How much fluid is actually being filtered at the glomerulus?
For that, we need a substance that is freely filtered at the glomerulus but is neither reabsorbed nor secreted by the tubules as it passes through. The classic substance used for this experimentally is inulin.
Inulin. Not insulin, inulin.
Correct, inulin, a polysaccharide. Because it's only filtered, the amount of inulin excreted in the urine per minute must equal the amount filtered at the glomerulus per minute. Therefore, the clearance of inulin gives us a very accurate measure of the glomerular filtration rate, or GFR.
GFR, that's the key measure of kidney function, isn't it?
It's the single best overall index of kidney function, yes. Clinically, we often estimate GFR using creatinine clearance, as creatinine is naturally produced by muscles and handled similarly to inulin, although not perfectly.
Right.
But inulin clearance is the gold standard for measuring GFR accurately. Typical GFR is around 125 milliliter.
125 milliliters per minute. That's like 180 liters a day being filtered.
It is an enormous amount. Of course, most of that gets reabsorbed. But these clearance measurements, RPF using PAH, GFR using inulin or creatinine, are absolutely vital diagnostic tools for assessing kidney health and tracking the progression of kidney disease.
Okay, can clearance also tell us if the tubules are reabsorbing or secreting something?
Yes, by comparing a substance's clearance to the GFR, measured by inulin clearance. If a substance is freely filtered like inulin, but its clearance is less than the GFR, it means some of it must have been reabsorbed back into the blood from the tubule.
Makes sense. Less came out than was filtered.
Right. And if its clearance is greater than the GFR, it means that in addition to being filtered, more of it must have been actively secreted into the tubule from the blood.
Like PAH.
Exactly like PAH. We can also calculate something called the fractional excretion, FE. That's simply the ratio of the amount of a substance excreted compared to the total amount that was filtered. FE, excreted filtered. or simply clearance of X divided by GFR.
So Fe less than 1 means net reabsorption. Greater than 1 means net secretion.
Precisely. It's a very useful way to quickly assess how the kidney is handling different electrolytes, for example.
Okay, but these clearance methods, they measure the whole kidney, right? Both kidneys working together.
That's a limitation, yes. They measure the overall function of roughly 2 million nephrons combined.
Mm-hmm.
They treat the kidney essentially as a black box. They tell us the net result, net filtration, net reabsorption, net secretion, but they don't tell us where along the tubule these processes are happening or the specific mechanisms involved within a single nephron.
So how do researchers figure that out?
To get that kind of fine detail, researchers have to use much more invasive microscopic techniques. The classic one is free-flow micropuncture.
Micropuncture sounds delicate.
It is. It involves using incredibly fine glass micropipettes to puncture individual nephron tubules or capillaries on the surface of an exposed animal kidney under a microscope. They can collect tiny samples of tubular fluid from specific known locations along the nephron or measure pressures and electrical potentials.
Wow. And what can they learn from that?
They can measure the concentration of substances like inulin and other solutes in the fluid collected at different points. By comparing the concentration in the tubule fluid, TF, to the plasma P, especially relative to inulin, which tracks water movement, they can figure out precisely where along the tubule water is being reabsorbed and whether a specific solute is being reabsorbed or secreted between different puncture sites. They can even calculate the filtration rate of a single nephron, SNGFR.
Incredible detail. Okay, so once the kidneys, through all these complex processes in the nephrons, have done their incredible work filtering, reabsorbing, secreting, the final product is urine. What happens next? Does its composition change much after it leaves the kidney?
Not really. By the time the fluid leaves the very last part of the collecting duct system, deep in the renal papilla, it is final urine. The remaining structures, the renal pelvis, ureters, bladder, urethra, don't substantially modify its composition or volume. Their job is transport and storage.
Okay, so transport starts with the ureters.
Yet the ureters take over. They act as conduits, basically muscular tubes, propelling the urine from the renal pelvis down to the bladder.
How do they propel it? Gravity.
Not just gravity. They use peristalsis, rhythmic, wave-like contractions of the smooth muscle in their walls. These waves usually start from pacemaker cells in the renal pelvis and travel down the ureter, pushing little spurts of urine along, typically at a rate of about 2 to 6 spurts per minute.
Okay. And how do they connect to the bladder? Is there a valve?
They enter the bladder wall obliquely at an angle. This clever arrangement means that as the bladder fills and pressure inside increases, it naturally compresses the part of the ureter passing through the bladder wall.
Ah, so it acts like a physiological one-way valve.
Exactly. It effectively prevents urine from flowing backward or refluxing up into the ureters and kidneys when the bladder contracts during urination. This is really important for preventing kidney infections.
Makes sense. Now, what if something blocks a ureter, like a kidney stone?
Ah, yes, that's a direct and painful clinical connection. If a kidney stone gets lodged and blocks the ureter, urine backs up behind it. This dramatically increases the pressure inside the ureter and renal pelvis, causing distension.
That sounds bad.
It is. This backup can lead to hydronephrosis, a swelling or dilation of the renal pelvis and calicities. And the distension itself, plus the often violent peristaltic contractions of the ureter trying to push the stone along, causes incredibly severe wave-like flank pain known as renal colic.
Ouch. Can damage the kidney.
Absolutely. If the obstruction persists, the high pressure can damage the kidney tissue and lead to a decline in renal function, even permanent damage if it's not relieved.
Okay, so assuming urine makes it down the ureters, okay, it reaches the urinary bladder. What's that like?
The bladder is essentially a hollow muscular storage tank for urine. Anatomically, it has a main body, which collects the urine, and a funnel-shaped neck that connects down to the urethra, the tube leading out of the body. It's lined by a specialized transitional epithelium that can stretch considerably.
And the muscle.
The bladder wall is primarily made of smooth muscle, arranged in roughly 3 layers, collectively called the detrusor muscle. When this muscle contracts, it squeezes the urine out.
Okay. And what about sphincters, keeping the urine in?
There are 2 main sphincters. At the junction of the bladder neck and the upper part of the urethra, the smooth muscle forms an internal urethral sphincter. This one is involuntary.
Involuntary.
Then further down, surrounding the urethra as it passes through the pelvic floor muscles, you have the external urethral sphincter. This one is made of skeletal muscle, which means it's under voluntary control. This is the one you consciously tighten to hold urine in.
Ah, the voluntary one. How is all this
Oh
nerves?
yes, intricate innervation. The bladder and sphincters receive input from all three branches of the peripheral nervous system: sympathetic, parasympathetic, and somatic, voluntary motor.
What do they each do, generally?
Very generally speaking, during the storage phase, sympathetic nerves tend to relax the detrusor muscle, allowing the bladder to fill, and constrict the internal sphincter, keeping the exit closed. Somatic nerves keep the external sphincter contracted.
So storage is sympathetic and voluntary control. What about emptying?
For voiding or mituration, it flips. Parasympathetic nerves become dominant. They strongly stimulate the detrusor muscle to contract, squeezing the bladder. At the same time, both sympathetic and somatic input to the sphincters is inhibited, causing them to relax and open the outlet.
Parasympathetic for peeing. Okay, how does the whole reflex work from feeling the urge to actually going?
Right, the micturition reflex.
Mm.
As the bladder fills with urine, stretch receptors in the bladder wall start sending signals up the spinal cord to the brain. You usually get the first conscious urge to void when the bladder holds around 150 milliliters or so.
Just a little bit.
Yeah. As it continues to fill, towards maybe 400 or 500 milliliters, that urge becomes much stronger, a feeling of fullness or discomfort. But normally, higher centers in your brain, in the cortex and pons, inhibit the basic reflex arc at the spinal cord level.
So you don't just automatically...
Exactly. You have conscious control. Voluntary contraction of that external sphincter also helps maintain continence when the urge is strong.
Okay, so then you decide it's time. What happens?
When you voluntarily decide to urinate, the first step is consciously relaxing your external urethral sphincter. Then, signals from the brainstem, specifically the pontine micturition center, coordinate the rest. This center inhibits the sympathetic outflow and strongly activates the parasympathetic nerves going to the bladder.
Triggering the detrusor contraction.
Yes, the detrusor contracts forcefully. This contraction itself stimulates more stretch receptors, creating a positive feedback loop that sustains the contraction until the bladder is pretty much empty. The internal sphincter relaxes passively as the detrusor contracts. Higher centers also ensure continued relaxation of the external sphincter. Sometimes, voluntarily contracting your abdominal muscles can help increase pressure and fully empty the bladder.
A coordinated effort. What happens if the nerves controlling this are damaged?
Clinically, this is very important. Lesions in different parts of the nervous system can cause various types of bladder dysfunction. For example, if the sensory nerves from the bladder are damaged, the person loses the sensation of fullness, the reflex doesn't get initiated properly, and the bladder can become hugely overdistended and flaccid, leading to overflow incontinence.
Just leaks out when it's too full.
Right? On the other hand, if there's damage above the main reflex center in the spinal cord, like in a spinal cord injury after the initial shock phase, the higher inhibitory control is lost. This can lead to a hyperactive or spastic neurogenic bladder, where the bladder contracts frequently and reflexively in response to even small amounts of stretch, causing incontinence and urgency.
Uncontrolled contractions.
Exactly. And in many of these conditions, the bladder might not empty completely, leaving residual urine behind. This stagnant urine significantly increases the risk of developing recurrent urinary tract infections, UTIs, which can be a major problem. It really underscores how crucial that intricate neural control is for normal bladder function.
It really does. What a complex and finely tuned system.
Absolutely. From the sheer volume filtered to the microscopic precision of the barrier and the coordinated control of storage and emptying, it's remarkable.
So we've taken quite the deep dive today into the urinary system. We started with that surprising fact about kidney blood flow.
Yeah, 20%.
Explored the kidney structure, the amazing nephron, the incredibly selective filtration barrier, the JGA control center, how the tubules process the filtrate, and finally the journey of urine out of the body. It's this system of just incredible filtering power combined with highly sensitive regulatory mechanisms all working constantly to keep you healthy.
And remember, as complex as it sounds when we break it all down like this, mastering this material is absolutely achievable. Taking it piece by piece, understanding the what and the why really does make it click. You're doing great just by digging into this.
Definitely. So, as you reflect on this, maybe consider this. How do systems like the urinary system exemplify this delicate balance between, say, robustness, that massive filtering capacity, and extreme sensitivity, those tiny adjustments in regulation? What happens when even a tiny part of that intricate balance is disrupted, like with that slit diaphragm protein or a nerve signal? And how does the rest of the system try, often remarkably, to compensate? It truly highlights the body's resilience and how interconnected everything is. You are part of the deep dive family, and you absolutely are capable of mastering this material.